There's like free printing sometime, and I was thinking about printing out, like changing the background to just white and printing out all of my notes for my friends for next year. You think they'll get mad at me, or I'll be like, I need it for my studying, I need it for class. All of it, it's cumulative.
Yeah,
Don't ask my
finals
professor.
week, I know there's free printing, but if you say it's cumulative.
Yeah, I need to look at my notes.
I think they do have a limit though, and it's only in black and white.
I go in over the course of like, God, there's 111 pages.
Oh my god.
I could put each page on like,
Just do 10 pages, like 20 pages each day. Maybe they'll let you do 20 pages.
I could put like 4 pages on a page.
Just shrink it all down.
Yeah, because I mean like the pages aren't.
Glass.
Yeah. Hey, it's fine.
Okay, so I did update the Canvas page to move lecture exam 4 content near the top, so it should be there visible now. It's more by this morning. And I went ahead and moved the sensory systems lecture content up there if you're watching the videos from COVID. And then the study guide is from last spring, and I think it's pretty accurate, but we'll check that again in next week. Because we have our lecture exam 2 weeks from today, right? That's our lecture exam is going to be 2 weeks from today. Our lab exam, last one's 1 week from today. So we're kind of coming up to the end here. Let's see, what else is there going on? I believe the lab exam 3 content is also visible there. So we'll take a look at that and closer to lab. Anyway, we actually really only have a couple topics left today and Tuesday for lecture content. So this is going to be kind of a short test, probably only about 4 pages long, and it won't be worth as many points as the previous test. So this one will probably be worth about 60 points or so if you're trying to estimate. That's good for you though, right? It's a short test, kind of easy. Okay, so let's go ahead and we're going to cover today respiration in vertebrates, and then we will cover circulation and osmoregulation on Tuesday. So I think we'll see what happens. I think today we will cover circulation today, and then we'll talk about it again on Tuesday with how the kidney functions or osmoregulation. But basically, we only have essentially this last topic, which is how vertebrates do their physiological adaptations for habitat. Okay, so primitively, we're looking at fishes as kind of a primitive example of a vertebrate, and from the lamprey to the sharks to the bony fishes, even the larval amphibians, they use what's called a pharyngeal pump in order to ventilate their gills. So this pharyngeal pump is they draw, let me go ahead and draw something on the board here. They draw water in, let's see, where is, go ahead and do a drawing here. We're going to show how this gill feature works. Here's our little fish. Okay. Beautiful fish. Okay. So anyway, what's going on here is they have a pharyngeal pump that draws water into their mouth and then basically out their gills. So make sure that, you know, when we talk about, or when you're describing this, you specifically say pharyngeal pump as a primitive feature here. Anyway, what's interesting about how this works is the water going over the gills actually goes over the gills in a very specific way. So how the gills are kind of oriented, and many of you saw this in the shark, when we were cutting the heart open to the shark, you saw the gill filaments were kind of oriented in a very particular fashion. Let me see if I can get this drawn correctly here. So the gills are basically here, you know, in the under the operculum. So let me go ahead and draw how those gills are kind of arranged here. Okay, so the gills are arranged in kind of like these, the technical term is lamellae, but what that really means is fingers. The gills kind of have these fingers sticking off of them. And so, looks something like this. Okay, so that's what kind of the an individual gill feature or the gill fingers are. There's like, you know, thousands of these basically on an individual gill. How that works is there is a artery that comes directly from the heart. Basically, the heart pumps. We'll cover circulation here at the end, but the heart basically pumps blood directly to the gills. But note that blood that's pumped to the gills is lacking oxygen. So even though it is an artery coming from the heart, I'm going to draw it in blue because I want to indicate that this doesn't have any oxygen in it. So it's deoxygenated blood. So this is it's called, let's go ahead and use this term. It's called the afferent artery. And what afferent means, and we'll see this term again with the kidney, is this is a blood vessel that approaches a capillary bed. And how I remember this, or you know, I still remember it this day, is the letter A you can think of as approach. So afferent basically means a blood vessel, usually an artery, that approaches a capillary bed. Okay, so how that works is that blood vessel goes down the outside of the lamellae and then essentially crosses over in the capillary bed of the gill. On the opposite side, basically the blood is picking up oxygen on its way across, like this. And then we have what's called an efferent. Artery. So these are both arteries because they're basically blood vessels being carried away from the heart, but the term afferent and efferent refer to whether it's approaching the capillary bed or whether it is exiting a capillary bed. And so you can remember the difference based on the A versus the E for approaching versus exiting. The technical term is afferent versus efferent. There's a similar system on the opposite side, but I'm just not going to draw it. So this basically is looking at one finger here. But there would be a parallel system here on this side as well. Got it? So I'm just not going to cloud it up. Okay, so what's interesting here is the water has a very particular way that it flows through this system. Basically, the water that's flowing over the gills goes in a counter-current fashion. So the water that's basically coming in here passes over the gills in a counter-current fashion. And that is important because it maximizes how much oxygen can be transferred. So let's go ahead and take a look at a close-up of this here. And we'll see how it looks. So what happens here is we have the afferent artery here. Something like this. So the blood's coming in here, afferent. And then, we'll go ahead and show it going out as the efferent. This. Okay, so what's going on here is the blood is coming into this side of the gill, and the blood is crossing over the capillary on the gill, and as it does, it's of course picking up oxygen along the way, and so the red is indicating oxygenated blood. The water is traveling the opposite direction, like this. Of course, when it comes into the gill, the water is fully saturated with oxygen, and as it passes across the gill, it's transferring oxygen into the blood vessel. So what you can say here is that as the water comes in, it has 100% O2 saturation, meaning it has the maximum amount of oxygen that the water can hold. Remember, cold water holds more oxygen than warm water, so we're just going to say that it's saturated for whatever temperature it is. And then as the water passes across the gill, it basically loses oxygen along the way. Let's see here. Make sure I have my numbers are kind of correct here. Yeah, that's cool. That way. We'll go 80%, 60%, 40%, 20%, and then it leaves usually at about 10 or so percent saturation. So this is basically the amount of oxygen that is remaining in the water as it passes across the gill. And then of course oxygen is being transferred to the blood, but the blood actually has an accumulation of oxygen that is a little different. So as the blood comes in, basically it has 0% oxygen saturation, so it's basically depleted of oxygen. And when it first enters into this kind of cross capillary, it begins accumulating oxygen from the water. And so oxygen is diffusing into the gill because the diffusion gradient is, there's a higher percentage of oxygen in the water than there is in the blood. And this basically happens along the entire length of the capillary here. And then when the blood basically exits here, it's like at 85 or more percent saturation of oxygen equivalent. So what this is called, this system that's really important to consider here, is this is called a counter current exchange. And in this counter current exchange, because the blood and the water are going in opposite directions, as the blood is coming in depleted of oxygen, the water is exiting mostly depleted of oxygen, but there still is the diffusion gradient where oxygen still diffuses across into the blood. And at every point along this capillary bed, there is a net transfer of oxygen to the blood side of the membrane. And so it kind of maximizes its efficiency, such that when the water initially comes in at 100% saturation, the blood is almost fully saturated, but there's still transfer of oxygen into the blood along that way. So this is kind of a really unique system. How they figured this out was, like for example, they would take sharks and they would actually pump water the wrong way over their gills. So if you put a shark in an aquarium, kind of hold it still, and rather than forcing water through its mouth, if you push water through its gills out its mouth, they found that the blood oxygen concentration drastically decreased. So even though they were exposed to water, if you had water going in the same direction, which is called concurrent flow, you would only get maybe about a maximum of 50%. So let's go ahead and use my favorite organism to compare everything to, the squid, right? Squidology here. The squid is a good example of what's called a concurrent exchange. And this is kind of how this works. The squid's not really concurrent exchange, but what it is if you remember how this works, is in the squid, they had a mantle cavity, and in that mantle cavity, they had these, let's see here, they had these cnidia that were just kind of hanging in the, mantle cavity. Remember, it had a circulation system. So anyway, what happens is water comes into the mantle cavity at 100% saturation, and then exits out after transferring oxygen to the gills. But because there's no specific direction of this, it's basically the gills are being bathed in water that has oxygen in it. Water comes in at 100%. The blood comes in basically at 0%. And in the blood exchange, basically, the max you can get would be an equilibrium, or a 50-50. So what happens there is you have water coming in at 100% O2, blood coming in at 0% O2, and when the blood and water basically are closely aligned at the gill, the blood will absorb the water, the blood will absorb the oxygen from the water, but basically what happens is at some point in time there's essentially an equilibrium where the water's lost half of its oxygen and the blood has gained half that oxygen. And so the maximum you could do would be about a 50% transfer of oxygen in what you would kind of call a concurrent exchange. This squid example isn't really a concurrent exchange. It's basically just the gills not having any particular direction or orientation in the circulation. But basically the idea here is that this counter-current exchange maximizes the efficiency of transfer. In this case, it's oxygen, but there's more examples that we're going to see in the kidney. A concurrent exchange also maximizes transfer of nitrogenous wastes and reclaimed water as well. So anyway, so on the exam, in addition to asking about respiratory evolution, which we're going to cover here more in a second, there'll be a question about the advantages of a countercurrent exchange system versus a concurrent exchange system. So, be sure you're kind of aware of that. The book has a decent description of this in the chapter on respiration, but this is kind of a simplified version here. The main points I'm looking for is you show the blood flows one direction and the water flows the opposite direction, which is why it's called countercurrent, and along the entire path you have a net transfer of oxygen from the water to the blood. Even where the blood is almost fully saturated, there's still a transfer that goes on there. Kind of the main thing. And then when I ask you to contrast it with like what happens in a squid, it seems like every test I've had that question. The thing to note here is because there's no specific orientation of flow between the blood or hemolymph and water, the best you can do is about a 50-50 or a equilibrium exchange between the water and blood. Okay? Sound good? Okay. So anyway, the next thing we're going to consider here is how vertebrates colonize the land. And so, I mean, obviously we looked at this with the amphibians, which would probably have been the first colonists back in the Devonian, that they have some sort of a lung or a respiratory pouch they use to do gas exchange. And what's interesting about this is that the respiratory pouch in amphibians and in other vertebrates as well is associated with the pharynx. So amphibians are using a pharyngeal pump to ventilate their lungs, to bring air into the lungs to do gas exchange. So they're essentially using the same pharynx mechanism that the fishes use to pump water over the gills, but they're basically using their pharynx to push air into their lungs. So when you see like frogs or whatever doing their inflating their throat or doing their croaking thing, what they're actually doing is they're kind of pushing air into the lungs very rapidly using their pharynx. Okay, so this basically begs the question of where did lungs come from? Or, you know, basically, what was the origin of the evolution of lungs? And so if we look back on our fishes that we looked at with the bony fishes, I had mentioned then that the bony fishes look something like this. They have a swim bladder. Draw a fish here. Okay, here's our fish. And what fishes have basically is associated with their digestive tract. Stomach here, intestine like this. So here's their gastrointestinal tract. They actually have a swim bladder that is a extension off of the pharynx, or right where the pharynx hits the esophagus. So this is called a swim bladder. Swim bladder, and it's basically the fishes can use this to collect air so they can regulate their buoyancy. Now this is the kind of complicated part. In primitive fishes like catfish or carp or garfish, for example, there is this little connection between the swim bladder and the pharynx. And this is called a neustonic tube, or it is basically a air tube that connects the pharynx to the swim bladder. In more derived fishes like trout, salmon, and swordfish, there is no longer a neustonic tube. They basically have separated the gut tract from the swim bladder, and the swim bladder is actually regulated by the dorsal artery. The dorsal artery will actually add or take away air in that swim bladder using essentially like an arterial pressure. This is why if you go deep sea fishing for rockfish and pull them up out of the deep ocean, like from 150, 200 feet or so, usually the swim bladder explodes. And you can see this in images. We'll pull up an image here here. So here's like a rockfish that's been pulled up from a couple hundred feet, and when you pull them up, basically the swim bladder, because the pressure is lower as you pull them up, especially out into the atmosphere, the swim bladder gets larger and larger and larger as you reel them up fast. They have no way to burp that air out, and basically it kills them. So when you're pulling fish up, you can even see the eyes bulging out because of the pressure difference. But because these fish don't have a connection to their swim bladder, they can't actually spurt the air out. More primitive fishes, though, like I said, like catfish, they can actually go up to the surface, gulp air, to do respiration if they're lacking air, oxygen in the water, but they can actually push air into their swim bladder to regulate that or spurt the air out. So there is this note that that primitive fishes basically have the connection, and more derived fishes don't have the connection. So anyway, this tube here, along with swim bladder, basically inspired many biologists in the 1800s, like 150 years ago. Many biologists were basically sharing the story that clearly the swim bladder was the origin, or the lungs are an exaptation of a swim bladder. So the idea here is that if you're looking at, say, for example, an amphibian, let me draw a frog here. Try to get a decent frog. This. Not too bad, frog. Okay, so here we have our frog, and they have a similar situation, which is they have a pharynx, and they have a gut tract. This, and their lungs basically are a paired feature that's on the ventral side of the gut tract. But lungs basically, seemingly, are essentially the same kind of pouch off of the pharynx where it meets the esophagus as the swim bladder is. So, Darwin, in Origin of Species, in 1859 or whatever, when he published this, all the way to the last edition, tells a story basically that it's pretty clear that fishes being primitive have a swim bladder, and that had been converted into or essentially led to an exaptation that was the lung. So this was Darwin's description that essentially lungs are adapted from a swim bladder. So that's, again, like the giraffe story. Giraffes reach high up in the trees to get food and out to their competitors. It's a real convenient story. But the problem is that it doesn't really make sense when we look at other primitive types of fishes. So let's take a look at this group that was discovered in the 1930s. And this is probably one of the most primitive bony fishes possible. I'll call it a coelacanth. So the coelacanth is a lobe-finned fish that, here's one with a diver here. The coelacanth is a lobe-finned fish that's found like in the Indian Ocean and the oceans around the Philippines, etc. This is considered kind of like a living fossil almost. It's a very, very primitive marine fish that doesn't actually have a swim bladder. It has essentially like what sharks have. It has a fat storage they use for buoyancy like sharks do. They don't use swim bladders at all. They basically collect fat or oil in a bladder or bag, kind of like the sharks use their liver for a semi-buoyancy feature. So the fact that our most primitive bony fishes that we know don't even have swim bladders. And then if we look at other lobe-finned fishes, these are fishes that have fins that kind of resemble stumps rather than rays, like we have with our bluegills and trouts, etc., have more spiny rayed fishes. Other examples of lobe-finned fishes would be lungfish. And so it was thought that maybe, here you can see the lungfish similarity to to a coelacanth. Basically, lungfish, there are several species in Africa and some in South America, but these fishes basically are also lobe-finned fishes, not ray-finned fishes, and they have a swim bladder that actually more resembles lungs than it does swim bladder. And what these fishes do is they use their swim bladder / lungs to move from pond to pond. So they can kind of crawl across land temporarily. So if we look at lungfish as an example here, these are fishes that, let me go ahead and draw a lungfish here. These are, again, lobe-finned fishes, like the coelacanth is. But these lungfish have a swim bladder that is ventral to the gut tract like the lungs are, and they use this for respiration. So anyway, the big mystery then is it was thought that essentially that a swim bladder turned into, let me go ahead and change this a little bit. I had bad planning here, but let me change this Darwin story here, was that the idea originally by Darwin was that this would be the progression. You'd have a swim bladder fish led to a lungfish, which led to lungs as being kind of like the final feature of moving terrestrial. So in the 1930s, when the coelacanth was found, and then more lungfish species were found, this has kind of been revised or sort of turned on its head. And the notion now is that probably more so, the oldest of fishes, like the coelacanth, didn't have a swim bladder, and swim bladders probably appeared as a result of respiration. It was actually a lung feature first. So if we consider the coelacanth, which doesn't have a swim bladder, the idea then is that the coelacanth led to fishes that had this bag, probably originated as a respiratory feature, and then the lungfish, being kind of a primitive fish, about a dozen species that still exist, but they were more prevalent back in the Paleozoic, led to two different possible options. Is that it's probable that the respiratory feature of this happened first, and the swim bladder feature was actually the later adaptation. So this is the current idea, meaning that today, the idea that lungs evolved from swim bladders is actually not correct. The idea is that what happened is fishes evolved a respiratory bag first that led to them being able to move on to land as amphibians and later, evolving into other things. And at the same time, fishes that stayed in the water converted that lung feature into a buoyancy bag or a respiratory feature. So on the exam, you're going to see a little paragraph snippet from Darwin's Origin of Species describing how the swim bladder neatly evolved into lungs, allowing vertebrates to colonize land. What I'm going to ask you is, what is the current story, or what's the current narrative of how lungs evolved? And actually, lungs probably evolved first as a respiratory feature for shallow water fishes like lungfish, and that the swim bladder that we know today is probably a more advanced feature than lungs, or came after lungs. So it's really kind of interesting how we can have a story being told for so long, for almost 100 years, from Darwin to the 1930s, and then by about 1950s or 60s, it got turned around that probably the lung or the respiratory feature of a lungfish probably evolved first, leading to terrestrial lungs in amphibians and at the same time buoyancy structures in waterfishes. So this is a lungfish. So anyway, that's an interesting article, but basically lungs would be our feature that we see for respiration in terrestrial organisms. And the main change that happens is what activates the lungs or innervates the lungs. So in reptiles, some reptiles still use pharyngeal pumps to activate that. For example, like turtles will use their pharynx, they'll use their throat muscles in order to fill their lungs with air. But organisms like alligators or large lizards like iguanas or water lizards actually use their rib cage, their thoracic muscles are used to basically expand or contract their chest cavity. So the pharyngeal pump is still used in some primitive reptiles, but ventilating the gills basically is transferred over to either rib cage muscles, or in the case of mammals, you'll see this today with the rats, is there's a diaphragm, a muscle that basically separates the peritoneal cavity from the pulmonary cavity. And when that diaphragm contracts, it basically increases the volume of the pulmonary cavity, ventilating the lungs. And in birds, they've evolved basically independent air sacs. Birds have a hard lung that's kind of a central hard lung for respiration, and then they have a number, about a half dozen air bladders that they use to circulate air over this hard lung. So the three different groups, the reptiles use their rib cage muscles, the mammals use a diaphragm, and the birds use air sacs. But basically, they're all derived from the same kind of lung feature, which was originally a pharyngeal type pump. Okay, so we're going to see the diaphragm today in the rats. I don't have any birds to dissect, so I won't ask you anything about the bird respiration or reptile respiration. But just kind of basically, we're going to think about the pharyngeal pump as kind of being the key feature. Okay, all right. So anyway. One thing that is crucial for any of this to work, for example, a countercurrent exchange or being able to get respiration from a lung, is that the heart had to actually evolve in order to transfer oxygen or transfer blood effectively to these organs. So what I want to do here is cram in a little bit about the 2-chamber versus 3-chamber versus 4-chamber hearts that we have seen or we're going to see today, the 4-chamber one with the rat, and talk about circulation as it relates to lung respiration. So let's go ahead and let's see, I'm almost out of room here, but I don't think that's too bad. I'll go ahead and squeeze it in over here. I know it's not much room, but we'll see what we can do. And I'll probably reflect on this again on Tuesday, so if it doesn't make any sense. So when we looked at the shark had, let's go ahead and look at our shark feature or our fish feature. So remember our shark here. Let's see here. Shark. Okay, it's a short shark, so I have my room here. The thing to remember with the shark is they had a 2-chamber heart design. Basically, their heart was a primary pump that pushed blood over the gills and then distributed the blood throughout the body. So what's happening here is the ventricle, the single ventricle, collected the blood from the venous system, and so there was a single atrium on the backside of the heart. Okay, so the atrium received blood from the body, deoxygenated blood, and then transferred the blood to the ventricle, and the ventricle was pushing the blood over the gills, and then distributing oxygenated blood to the rest of the body, the carotid arteries and then the dorsal aorta. So if we're going to look at this design, how this is set up, this two-chamber heart design, there is basically a single ventricle, and we're going to look at the shark heart basically here as an example. And with something like this. Okay, so there's our ventricle, V for ventricle, and then here is our atrium behind it. And then here is our sinus venosus, which is basically the major collection of all the veins. Sinus venosus. So that's kind of a two-chamber design. It's like the shark and the fish basically have the same kind of two-chamber design. So when the blood comes back from the body, it goes to the sinus venosus, then it goes to the atrium. The atrium pumps the blood effectively into the ventricle. The ventricle contracts, pushes the blood out of those arches to the gills, and that's where respiration happens. Let's also reflect on which gill arches these are. This is gill arch number 7, 6, 5, 4, and 3. These are the basically gill arches. And I mentioned that gill arch number 1 and 2 have been converted into jaws, right? So the mandible arch and the highway arch are no longer for respiration. They've lost their circulation connection. If I drew a lamprey heart, they would have 7 arches, right? They would have all 7 of those arches. That's kind of what's going on with the circulation system. What's really interesting is that if you think about it, the blood coming into the heart from the body has no oxygen. It's deoxygenated and does not pick up oxygen until it travels through the gills. As a result, these organisms have to have a separate system to return oxygenated blood back to the heart. And this is what's called the coronary artery. And it's basically a result of the heart pumping deoxygenated blood to the gills to pick up oxygen. We have to return oxygen to the heart so the heart can pump and do its action. Very, very high metabolic demand. So there is a coronary artery that comes off right behind the gills. We didn't really see where it came off on the shark, but I think on the shark's heart you saw the coronary artery probably, or some of them are injected. But that's an interesting ancestral baggage that we're going to see even in the 4-chamber heart. Even though half of your heart has fully oxygenated blood from the lungs, because it's lined with endothelium, your heart can't actually get any oxygen from that blood. You also have a coronary artery that comes from your dorsal aorta, and it supplies your heart with oxygenated blood. And so this feature kind of travels through the amphibians and the mammals as well. Okay, so then we're looking at, this is a 2-chamber design, 2-chamber heart. 2-chamber heart, which would be found in fishes, cartilaginous fishes and bony fishes. When we get to the amphibians, we now have a system that requires getting blood to a lung system. So let me go ahead and draw what the amphibian three-chamber heart looks like. We saw these. We saw this in our dissection. We didn't really see all the branches, but there are 3 branches. I think I drew it last time also in the lab. Okay, so there's 3 branches in the three-chamber heart, and then there are 2 atria. There is a right atrium, and there's a left atrium. Okay, and then there is a common ventricle. Okay, and then this is a 3-chamber design found in amphibians, lizards, and snakes. Will have a 3-chamber heart. So anyway, one interesting question is, where did the other atrium come from? It's likely, if you were to imagine this whole thing twisting, the right atrium basically comes from an origin that's probably what the sinus venosus was. The right atrium is actually receiving blood from the body. This is like the posterior vena cava, and this is the anterior vena cava. But basically the blood comes back from the body to the right atrium, and then the blood is transferred into the ventricle from the right atrium. When the ventricle contracts, it sends blood out all 3 branches. These are parallel branches. This branch basically goes to the head. It's called the carotid arteries, but it's head and forelimbs. This branch goes to the abdomen and hind legs. And then the final branch here goes to the lungs. So this is the pulmonary, or lungs. So, and if we want to say what number arches these are, I think I was showing you with the frog or the salamander skull that has the gills still on it. Basically, this is gill arch number 6, 5, and 4. Essentially, what we can say is the 7th gill arch is vestigial; it's gone. The 3rd gill arch had turned into the operculum in the bony fish, and so what we're left with basically here is 4, 5, and 6 as our gill arch. And gill arch number 6 now is still a respiratory arch, but rather than sending blood across the gill, it's sending blood to the lungs to pick up oxygen. And then the blood from the lungs comes back to the left atrium. But what's interesting to me is that the left atrium now dumps oxygenated blood back into the ventricle, along with deoxygenated blood that comes back from the body. So the ventricle basically has a mixture of body blood, low in oxygen, and lung blood, rich in oxygen, where that mixes together again and it gets sent out basically to all 3 circuits, the head, the abdomen, and back to the lungs. So it seems like this doesn't really make any sense. Why would you send blood to the lungs to get oxygen, only to bring it back and mix it with blood that came back from the body? But I think I mentioned with the frog dissection that amphibians and even some organisms like turtles can actually do quite a bit of their respiration across their body surface. So the blood coming back from the body really isn't 0 oxygen. It actually has oxygen that came from cuticular respiration across the skin. But of course, in large toads, or of course in reptiles, like a monitored lizard or a iguana or something, they don't really have cuticular respiration. So there are mechanisms by which those organisms can actually partition the body blood versus the lung blood. And I'll mention that in a second here. But the interesting thing about three-chamber heart is the ventricle basically is a mixture of right atrium, low oxygen blood, and left atrium, high oxygen blood mixed together. All right, finally, and we're gonna see this in the rat today, is we have a 4-chamber design. It looks something like this. Question? Oh. We have a 4-chamber design. And this is what's going on in the 4-chamber design. Okay, 4-chamber design is kind of like what the rat has. Okay, so it's kind of a different plumbing here. Let's see here. All right, there is our ventricle, and the ventricle is divided into 2 halves. So we have our left ventricle, and we have our right ventricle. The same ventricle, but it basically has a septum that separates the ventricle into 2 compartments, left and right. The atria are in the same spot, and they actually serve the same function. The right atrium receives blood from the body, prevena cava, and posterior vena cava. In humans, we call it superior and inferior when we stand up. But basically, this is from the jugular veins, and this is from the cardinal veins of the body. And then the left atrium is receiving blood back from the lungs. Okay, so in this system, which is pretty fascinating, is blood comes back from the body pretty much depleted of oxygen. So there's no oxygen basically coming in to the right atrium. That blood then is transferred to the right ventricle, and then from the right ventricle it goes out to the pulmonary system. So this would be our pulmonary circuit. Let me go ahead and use green a bit before. This is to the lungs, or a pulmonary circuit. So the right ventricle basically receives low oxygen blood from the body. The right ventricle pumps that to the lungs, and then the blood comes back from the lungs loaded with oxygen and goes to the left atrium, where it is transferred into the left ventricle. The left ventricle then sends this fully oxygenated blood out to the rest of the body. So it's getting kind of complicated. Anyway, what's interesting is the question might be like, well, where did all this come from? Where did this extra chamber come from? And why is this arch so weird? Well, really what's going on here is, if you think about it, if you were to run a dividing line up this ventricle, you could actually say that's what happened here, is there's a dividing line that runs up here, and that the pulmonary circuit is actually arch number 6. And what we could say is, if we divided this up here, we could say that this arch number 6 is basically our artery that goes to the lungs, the pulmonary artery. And then the pulmonary vein comes back to the left atrium, and that here we have what goes to the head. So these are 4, and then this one is 5. So what's going on here is essentially we've kind of modified the heart, that here we have 4 and 5 are delivering blood to the head and the body, and arch number 6 delivers blood only to the lungs. And it's because of this septum that goes up through the middle of the ventricle that this separation happens. So this is what a mammal heart would look like, a 4-chamber design. This is our 4-chamber heart, because it has because it has basically 2 atria and 2 ventricles. Interestingly enough, birds and alligators and crocodiles also have a 4-chamber heart, but theirs is built backwards. That they have a aortic arch like this, but it goes the opposite way. It goes to the right rather than to the left like it does in ours. In mammals, the arch goes to the left. In birds, it goes to the right. So it's likely that birds and crocodiles and alligators evolved a 4-chamber heart separately or independently from mammals. This is kind of built backwards, like a story we talked about with embryology. And interestingly enough, like I said, there are some large lizards like Komodo dragons and monitor lizards that have a partial separation or a septum that partially divides the ventricle. So you could almost say that that would be like a 3.5-chamber heart. That septum can partially segregate oxygen versus deoxygenated blood. And to put that in comparison is our last comment here. In mammals, because basically the fetus is being carried in the womb and being fed oxygenated blood by the placenta. So the placental connection basically is supplying oxygen to the developing fetus's blood system. So in that case, the lungs are not really being functional while the fetus is developing in the amniotic sac or fluid. So babies, human babies, just before they're born, still have an opening or a connection between the left ventricle and the right ventricle. And the kind of developmental planning is just before you're born, this gap is grown over and separates both the right and left ventricle. And you might hear about people who have pregnancy complications where the baby's heart's not fully developed. They can actually determine if that has been sealed up or stays open. And pretty amazing that they're able to actually do heart surgery on babies just before they're born if they don't have that complete. But the fact that babies before they're born still have an opening kind of implies that there is a relationship between a 3-chamber heart and a 4-chamber heart. Basically a septum grew up the middle to divide the two. And that division that happens basically just before mammals are born is kind of like a remnant or a ancestral baggage of having that design. So anyway, I think for our purposes, I would like you to, on the exam, compare and contrast a 2-chamber heart, a 3-chamber heart, and a 4-chamber heart. I know these diagrams are really messy, so the main thing I'm looking for is that you note that the 2-chamber heart basically has a ventricle and an atrium, along with a sinus cavity to collect from the veins. The 3-chamber heart has a single ventricle with a right atrium and left atrium. The right atrium receives blood from the body, left atrium receives blood from the lungs. And then the 4-chamber heart design basically fully segregates blood from the body, sends 100% of it to the lungs before it's returned and delivered back to the body by the aortic arch. But the main thing I want to point out is that these are basically all homologous. Essentially, as vertebrates colonized land and became more metabolically demanding, especially with being warm-blooded like mammals are, they had to basically develop a system that had as efficient of oxygen transfer as possible. Okay, so plan on just an easy question on compare and contrast a 2-chamber, 3-chamber, and 4-chamber heart. Maybe list which kinds of organisms have each. This would be the fishes, this would be the amphibians, this would be the mammals, and that'd be a straightforward question there. Okay, I know we went a little long in lecture today, but let's take a longish break, like 15 minutes, because I have to euthanize our rats. The rats are fresh, and that's great because you can really see the tissues and the pliability. I don't really care for all these preserved things that we've done because it's kind of hard to see the real structures. But that's why I tend to love the near live rats, because you can really see the features well. So I'm going to go ahead and euthanize those. I'll erase the board, we can blank it back, and I'll draw out what we're gonna see in the graph. All right, so quarter till. Let's go ahead and take a break for that long.